Hello, my name is Mivo Biju, a student at Thomas Jefferson University's East Fall campus. We will be looking at a study on T4 phage specificity for infection in E. coli and not in B. cereus. In this slidecast, we will be looking at the introduction, hypothesis, results, and conclusion of this phage specificity lab. Bacteriophages are viruses that infect bacteria. However, these viruses are limited by their host range. This means they can infect only a specific number of cells within specific species. In this experiment, we will examine the specificity of coliphage T4 with the bacterias E. coli and B. cereus. T4 phage is a bacteriophage, a virus that infects bacteria. It is only capable of undergoing the lytic cycle as seen here. After the viral DNA is injected by the tail, synthesis of the viral genome and proteins is conducted with the aid of the host cell's resources. Once the new phages have been created, the host cell is split open to release the phages. E. coli is an anaerobic bacteria that is found in the human body and helps with digestion. Certain versions of E. coli can cause gastrointestinal illness and is contagious via stool. B. cereus is an anaerobic bacteria that is typically found in soil or plants. It produces toxins which, when consumed, can also cause gastrointestinal illness. So the hypothesis for this experiment was if T4 is specific only for E. coli, then there should be holes or plaques in the agar plate with both E. coli and T4 phage because T4 lysed and killed the E. coli cells. These are the negative control plates, which are just the bacterial strains on the agar. The negative controls are the groups which receive no treatment and thus should not exhibit any changes. These two plates have the bacteria in the presence of the T4 phage. On the left there is B. cereus with T4, but there is no change in the plates such as the presence of plaques. Meanwhile, on the right is E. coli infected with T4. As observed, there are many plaques scattered throughout the plate. Overall, two of the agar plates consisted of only top agar and either E. coli or B. cereus culture. The other two plates consisted of top agar, the specific bacterial culture, as well as the diluted phage suspension. Solidifying and incubation took place to allow the host cells and phage plaques to form. Only the plate with both E. coli and T4 showed any variation. It produced approximately 90 plaques throughout the plate, signifying bacterial death due to the T4 infection. The absence of bacterial cell death in the B. cereus plate as well as the control plates of B. cereus and E. coli tells us two things. One, that T4 cannot infect B. cereus due to lack of specificity and recognition. Secondly, that the control plates tell us the obtained results are due to T4 and not to any other external factors. It can be concluded that T4 has specificity for E. coli and lacks recognition for B. cereus. The hypothesis for this study, that T4 is specific to E. coli and thus will cause death by lysis, as observed in the agar plate, was supported by quantitative observations from this study. After examining external studies, it seems that this experiment's results coincide with others. Specifically, the research by Vanderbilt's Department of Medicine, which examined the attachment of T4 to E. coli. Amongst various other factors, this study showed that T4 absorbed to bacteria by tail fibers. These tail fibers are crucial to the small host range and specificity of T4 to the E. coli bacterial cell. We can apply our results to develop medicines to treat E. coli and other bacterial diseases through bacteriophages. Moreover, by applying a similar procedure, we can understand other bacteriophage specificity as well. This is a list of the works referenced and cited in this slidecast. This is a list of the images cited in this slidecast. Thank you for watching.